Great. You guys are coming back on. Can you guys hear me now?
I can hear you.
All right. I don't know what happened. It had the microphone on, and for some reason you guys couldn't hear me. So Zoom just stopped working, I would assume. But good, you're still here. So let's resume the meeting. That means there's going to be two Zoom sessions for this lesson. My computer is going to be slow because it's currently making the file for the first Zoom meeting. And let's get rid of that and back here. And I got to share my screen. Whoops, don't want that. All right. All right. So there's two ways sexual conjugation can happen. That's with the sex pilus in the gram-negative cells, and then with the mating bridge in the gram-positive cells. In the mating bridge, what happens is two gram-positive cells come together. They're usually two cells of the same species. And then the cell membrane fuses. So the cell membrane doesn't go like that. It actually comes around and then like that. And so the cytoplasm of this cell can freely mix with the cytoplasm of that cell. DNA from that cell can leave this cell and then enter that cell, or vice versa. And that is how a mating bridge allows sexual conjugation to happen, where DNA can move out of one cell and then into the other cell. Usually, when the DNA is moving from one cell to another cell, the plasmid, it's usually a plasmid, will replicate so that the copy of the plasmid will move out of this cell and into that cell, or move out of that cell and into that cell. And that way, both cells now have that plasmid. Any question about any of that? So let's look at sexual conjugation with the sex pilus in a little more detail. The sex pilus will go out and skewer. Good, my computer's not going to speed up. Will go out and skewer an F minus cell, meaning the sex pilus is created by the F plus cell. And then it'll act as a winch to bring the two cells together. They do not have to be this close together because that sex pilus is hollow. And then the F plasmid right here will replicate and then send one copy of it into the F minus cell. That's important because the F plasmid codes for the creation of the sex pilus. And now the F minus cell has the plasmid in it, so this cell will grow a sex pilus, and then this cell will not be an F minus cell, it'll be an F plus cell, and it'll go uh, engaging in sexual conjugation with an F minus cell. So it changes the phenotype of this F minus cell, then becomes F plus cell. Any question about sexual conjugation? All right. The third way that um, genes may move around in a population is by transduction. Transduction happens when a virus moves genes from one cell to another cell. I realize we haven't talked about viruses yet, so let me introduce the topic so I can talk about transduction in bacteria. In bacteria, when a bacteriophage, meaning a bacteriovirus, infects the cell, it will attach to the cell and then inject its genome into the cell. The first thing the genome of the virus does is degrade the genome of the host cell. Why? Because the genome of the virus wants to call the shots in the cell. And the easiest way to do that is to have your genome, the only genome running and capable of functioning in the cell. And so it degrades the host chromosome. You don't need to know this, but it secretes an exonuclease, which then cuts the DNA of the host cell. And then the genome of the bacteria uses the bacteria to replicate the virus. So first it will replicate the viral genome, and then it'll transcribe the viral genome to make the viral proteins, and then the virus will assemble, and then the last thing the virus will do is rupture the cell, and that's to release the newly replicated virions. Let me blow this up. But what happens if a mistake is made? Viruses aren't worried about mistakes too much because they make a lot of offspring. And if one of their offsprings is a mistake, it's not a problem for the virus because it's got lots of offspring. If you notice, the viral DNA is, I don't know, red purple color, and that's inserted into this virus. But look at that virus or that virus. It's inserting a blue purple DNA. The blue purple DNA came from the cut genome of this cell. Then, when this virus binds to a bacteria, it will, because the protein of the virus binds it to the bacteria. It'll then inject its genome into the host cell. But this is not viral DNA. It's DNA from this host cell. And then this DNA can incorporate into the chromosome of this cell and then be expressed by this cell. That can change the phenotype of this cell because a new gene or genes have been added from the DNA of this cell. So, for example, if this cell was sensitive to antibiotic resistance, and then this DNA had a region of it coding for antibiotic resistance, this cell will now become antibiotic resistance. Any question about any of that? Well, how important is that? Let me end the show here and show you. And how important is transformation? We already said transformation happens 10 times more frequently than spontaneous mutation. So does sexual conjugation, at least in gram-negative cells. It also happens in one cell for every 100 million cells. And then transduction happens at about the same rate. So, meaning transformation, sexual conjugation, and transduction happen 10 times more frequently than spontaneous mutation. Everyone see that. Transformation, sexual conjugation, and transduction cannot create a new genetic variant in the species, but it can shuffle the genes from one individual to another or from one population to another so that new genes can be brought into a cell from genetic material already existing in the population or in the species. If there's no genetic information, then transduction, sexual conjugation, and transformation cannot happen. And it has to happen only by spontaneous mutation. So if there's no genetic variation in the species. The only way it can be brought into the species is by spontaneous mutation. An example would be something like if humans ever get the yellow eye color, it will first have to happen by spontaneous mutation. Any question about that? Now, transduction does not happen very often in the human genome because you'd have to have a virus infecting a gamete of a person, and then the virus goes to another person's gamete and brings in the DNA. That's not likely to happen. However, if you look at our human nose cells, I will bet all the time that people are having transduction happening in your nose cell. But who cares? Because your nose cell, even if it's genetically changed by transduction, who cares? Because that cell will not be passed on to the offspring. But think about when you have a cold, you can have the possibility of transduction leaving your cell and then entering somebody else's cell, changing the DNA of their nose cell. So transduction probably happens in the nose cell, but in the human genome, it probably doesn't happen very often. Sexual conjugation is really specific to bacteria. We are a sexual reproducing species, which shares a lot more DNA between two individuals to have the offspring than sexual conjugation does. But in bacteria, sexual conjugation is important because genes can leave one cell and then enter another cell. And bacteria, if you don't know, they don't undergo sexual reproduction. And then transformation happens in bacteria, but it's not likely to happen in the genome cell of your gametes because the DNA would have to be in the environment, come into your gamete, and then recombine with your chromosome in your gamete. And for one thing, our DNA is going to be stopped by the skin, meaning if you're like swimming in a pool and you come across some DNA, it's going to be stopped by your skin. And then if it were to join one of your cells, it's not likely to join one of your gametes. So transformation does not happen very often in human cells, at least in the human population. All right, any question about any of that? In that. We've talked a little bit about plasmids. A plasmid is a small circular extrachromosomal piece of DNA in a bacteria or in a yeast. A yeast is the one eukaryote that does have plasmids. Most eukaryotes do not have plasmids. The difference between a plasmid and a chromosome is the chromosome has thousands or more genes on the chromosome. Could be tens or hundreds of thousands of genes on the chromosome. A plasmid only has a few genes on it. So that would be one gene, maybe one gene here. There's 3, 4, maybe 5 genes on this plasmid. A plasmid can also replicate independently of the cell's chromosome. And actually, we saw that earlier when we were talking about the F plasmid. Oh, wrong way. Right here. This plasmid right here is replicating. When one copy is going into the F minus cell, the chromosome is not replicating. So the plasmid can replicate independently of the chromosome. The main difference between the plasmid and the chromosome is just the size of the DNA, the number of genes. The plasmid is only a few genes, it's really small. The chromosome is very large with many, many genes. There are different categories of plasmids. That's all you need to know. You don't need to know the names of the categories or what the categories are. Just should know there are different categories of plasmids. Any question about plasmids? All right. A transposon is a separate genetic element that is different from all other genetic elements in that the transposon can move from one region of DNA to another region of DNA. A transposon always has a transposase gene that encodes for the cutting out of the transposon right here. And then it has inverted repeats next to the transposase gene. The inverted repeats help the transposon leave one region of DNA and then enter another region of DNA. The transposase gene itself cuts out the transposon right here and here, and then it cuts in another region of DNA to insert the transposon. So the transposase gene is an enzyme that literally cuts out the transposon and then cuts open the new region of DNA for the transposon to move to. Transposons are important because they shuffle around the DNA of a cell. So you can have a transposon move from one region to another, and now you have two genes close together that used to be far apart. It sort of shuffles up the DNA within the cell. And that's important because I guess I should step back and talk a little bit more about plasmids. Plasmids are very important for bacteria because a plasmid is much more likely to leave the cell and then re-enter another cell than the chromosome. So a plasmid allows for bacteria to shuffle the DNA between two different bacteria cells, and then the transposon allows the bacteria or any other cell to shuffle up the DNA within their cell. Complex transposons carry other genes besides the transposase gene. So this is the simplest transposon. Here is a more complex transposon that has the kanamycin resistance gene in the transposon. So this kanamycin gene can move around in the cell. And that's important because it could move from the chromosome to a plasmid, and then the kanamycin gene is on the plasmid, so it's more likely to move into another cell. A complex transposon always has a complete transposon on one side and then another complete transposon on the other side. So this transposon actually has two transposase genes, this one right here and that one right there. And then it has two inverted repeats, or I should say two pairs of inverted repeats. This inverted repeat here and there, and then this inverted repeat there and this one there. These inverted repeats, this one here and this one here, are important for the transposon to leave the region of DNA and then insert into the new region of DNA. Any question about any of that? All right, and this is showing you that the kanamycin resistance gene can move from the chromosome to a plasmid. And now this is plasmid is much more likely to be given to another cell than the DNA of the chromosome. So the chymomycin gene, resistance gene, can more freely move in the population of cells because the chymomycin gene has entered a plasmid. Any questions about transposons? And I guess plasmids. All right, if there's no questions, let's talk about genes and evolution. Genetic variation, for it to result for the first time in a population, it has to come about by mutation. But if that mutant does exist in the species, then it can move around between cells by transformation, conjugation, and transduction, with crossing over and transposon just mixing up the DNA, generally in the bacteria cell, creating new variants that can be brought next to each other. So transposition and crossing over mix up the DNA between the DNA of an individual or a cell, and then transformation, conjugation, and transduction can mix it up between cells of the same generation. And then both can happen by spontaneous mutation, where you can get it arising by spontaneous mutation, as well as spontaneous mutation in a little ways can create new genetic variants, either by crossing over or transposition or even without, just because you got a new gene here, and so it's going to be next to a different gene next to it. Anyways, all of that stuff together, meaning mutation, crossing over, transformation, conjugation, transduction, and transposition, all of it creates genetic variation of the genes in a population of cells. That genetic variation can then drive evolution, acting via natural selection, meaning natural selection can act on this genetic variation and then drive evolution. Any questions about genes and evolution? All right, that's it for chapter 8. Let's briefly get started with chapter 14. Come on. Oops, didn't mean to open that. Let me make sure I'm supposed to do chapter 14. Yep, chapter 14. Chapter 14, Principles of Disease and Epidemiology. This slide is showing you the major goals and a rough outline. You'll note I've only got 3 terms here, but if you learn all the terms of this chapter, you'll actually learn 50% of the material to this chapter, meaning the terms are really important in this chapter. So know what pathology is, etiology, and microbial antagonism. That's 1. 2, be able to understand the relationship between the host and its normal microbiota. So know what the normal microbiota is also. And then 3, understand the stages of disease development. Disease normally goes through certain stages of disease development. And then 4, understand what nosocomial infections are and how to control them. Any question about any of this? All right. First, a few terms. Pathology is the study of disease. Etiology, the study of the cause of a disease. For example, the etiological agent of anthrax is the bacteria Bacillus anthracis. Pathogenesis is the development of a disease. So if you get exposed to anthrax, you likely will go through pathogenesis where the anthrax disease will develop. Infection is the colonization of the body by a pathogen or the presence of a microbe where it is not normally found. For example, if E. coli is transmitted to your lungs, it is not normally found in your lungs, and this would be an infection by E. coli growing in your lungs. But E. coli is normally a part of your normal microbiota, and we have probably millions of them in your gut. So infection is the colonization of the body by a pathogen or the presence of a microbe where it's not normally found, like E. coli in your lungs. A disease is an abnormal state in which the body is not functioning normally. You should come to know that microorganisms are pathogenic, but only a few of them are pathogenic. Most microorganisms do not cause human disease. And some microorganisms actually can benefit the host, especially the cells of the normal microbiota. Not all of them, but many of the cells of the normal microbiota can benefit the host. Any question about any of that? All right, let's talk a little bit about the normal microbiota. It establishes a relationship with its host. There are large communities of microbes found in association with all animals and plants. These communities living with us and on us or in us are stated and collectively known as the normal microbiota, a microbe that essentially colonized you shortly after you were born and then stayed with you. So the normal microbiota is normally with you all the time you're alive. The normal microbiota is only found in certain regions of the body, and disruption of the normal microbiota and its host balance appears to be associated with disease and impaired immune function. What do I mean by that? Well, all of you have normal microbiota in your gut, and that normal microbiota has established a relationship with your host. So, yeah, it's a normal microbiota. But there's a host balance, meaning that if the normal microbiota were to grow in your gut too much, your host will, I don't know, prune it. So there's a balance between the normal microbiota and the host. Well, what happens if you take antibiotics? It will, especially oral antibiotics, it will get into your gut and kill a whole bunch of microbes. This will disrupt the normal microbiota host balance, and it can cause the patient either diarrhea or constipation. Fortunately, most of the time this happens. The normal microbiota host balance reestablishes itself, and then the patient quickly loses the disease or impaired immune function. Any question about any of that? All right, this picture is showing you the locations of the normal human microbiota. And I should mention that we do have a normal microbiota to every species organism, meaning all humans have about the same normal microbiota. That's not entirely true, but it's similar. And then cats have a normal microbiota, but cats have different species as part of their normal microbiota. For example, cats, talking about domestic kitty cats, but probably other cats too, um, they have Bartonella henselae as a normal part of their microbiota, meaning that bacteria grows in and on cats, Bartonella henselae, and it doesn't cause the cat any disease. So it's a part of its normal microbiota. But if Bartonella henselae is growing on a patient, they're likely to get a disease. And I used to know that the disease is cat scratch fever. So if you have Bartonella henselae growing in or on you, you likely have cat scratch fever. This slide is showing you that where the normal microbiota can be. The skin, obviously, the gastrointestinal tract from the mouth down to the anus, large number of bacteria, especially in the lower part of the gut. But we also have normal microbiota in our eye, in our mouth. I already mentioned skin, large intestine, or normal microbiota in our urinary and reproductive systems. The normal microbiota differs from the transient microbiota. In the normal microbiota, once it colonizes a person or a patient, that patient likely is going to contain that normal microbiota species for the rest of their life. Like most of us got E. coli shortly after our birth, or maybe even during our birth, and E. coli became a normal part of our normal microbiota, and we will have E. coli on our skin and elsewhere, generally speaking, for the rest of our life. And that's what the normal microbiota is, something that colonizes a person early on in life and then stays with them, and then they have it for most of their life. It differs from the transient microbiota, and the transient microbiota may be present for days, weeks, or months, but unlike the normal microbiota, it does not permanently become established. An example of transient microbiota I got when I went to visit my aunt. She insisted on hugging us when I arrived, and she actually had a Staphylococcus aureus skin infection. She probably thought it wasn't contagious because she was under antibiotics, but shortly thereafter, I tested my skin in the microbiology lab, and sure enough, I was growing Staphylococcus aureus for the first time I'd ever seen it. Fortunately, 3 months later in the next lab, I tested my skin again, and I never became positive to the bacteria I bought, not the bacteria, the pizza I bought. What am I talking about? I never had Staphylococcus aureus after that first time, after hugging my aunt. After that time, all the times I checked my skin, which generally was about every 3 months for several years, I never had Staphylococcus aureus again. So for me, for that brief period of time after hugging my aunt, I had Staphylococcus aureus as a transient microbiota. And fortunately, I'm glad it disappeared because if you have Staphylococcus aureus on your skin, and 20% of Americans do, it's more likely going to cause a boil. And patients who have Staphylococcus aureus growing on their skin are much more likely to get a boil than a person who does not have Staphylococcus aureus growing on your skin. Any question about any of that? If not, I'm going to end it here, and I will continue with this part of the lesson next time. Remember, in the lab, We're not going to have a lab. You only need to show up if you have questions. I will be there for 15 minutes to answer any questions. After that, I'll log out. All right. I'll see you in the lab.